Okay, Garry, we got your new presentation in PDF form, slides, and it's really to help people understand where they are with modern diseases, in particular autoimmune disease. Is that right?
Yeah, that's right. The presentation will give them that information, but in order to get there, first of all, we've got to tell a little bit of a story about how it's come about, how this new model of inflammation and disease has been developed. And in order to do that, we need to give some background information, first of all, on cells, inflammation, this way, a new way of thinking, and in order to basically explain this idea, I need to explain how it's developed from an understanding of cancer. Yeah.
Interesting. Okay, so we'll go through the slides then, and you talk us through each slide. Yeah?
Okay. So I'm moving on to slide 2 now, and I'll just give you an overview of the content of the presentation. So first of all, I'm going to give you a very quick introduction to an emerging new discipline called systems biology. I'm going to talk about cancer as a complex disease. I'm going to talk about cells, inflammation, and the immune system. I'm then going to talk about how the current way of thinking, which is known as reductionism in medical research, is basically not sufficient to deal with the complex diseases that we have at the moment.
Interesting,
yeah. Then going to move on to an application of systems thinking. Well, my application of systems thinking in cancer and how that thinking came about. Then I'm going to talk about this new model of inflammation and disease and what that actually means.
Yeah. Okay, that's infection 2. Yeah?
Yeah, oh absolutely. Yeah.
Okay,
yeah.
Well that's interesting. Let's get on to that.
Okay, so page 3. Systems biology as a distinct discipline was introduced by a Professor Mazoverish in 1966 when he launched this new way of thinking, which was basically combining systems theory and biology. But systems biology as a subject was really spurred on by the completion of the Human Genome Project around about the turn of the millennium, and the need to start thinking big across huge amounts of data, because basically the Human Genome Project came up with a huge amount of data about, essentially the coding for what makes up organisms, but the data there was just so much detail. You'd gone down into so much detail that the data didn't really tell you how biological functions work. And the data certainly doesn't explain to you, how people work, how you think, and it certainly doesn't explain, why we're alive. These properties, these functions are when you take lots and lots of smaller things, you build them up into a much bigger picture. And there's an expression: the overall system is more than the sum of the components. That's called an emergent property, and these functions and life itself is essentially emergent behaviour.
Now, so how your genetic makeup, how they interact, the genes interact, is a factor in disease.
It's certainly a factor, but the way that, we'll talk about that on a couple more slides, but another thing to bear in mind is something like 90% of the genetic code isn't actually used. It's redundant, it's not used, or it's only triggered to actually do something in non-normal circumstances. So the first institutes of systems biology were established in Seattle and Tokyo in 2000. There's now 6 systems biology research centres in the UK. And this is all really, it's very, very new. And it was all spurred on by this huge amount of data that they got. But what does it actually tell you? Because they don't know yet.
No.
They're still working it out. So traditional research in medicine has largely been brought about by a mechanism called reductionism. It's a way of basically breaking down a problem into smaller and smaller parts in order to understand what's happening, the nature of the problem.
Is this the slide now, new thinking required to deal with complexity?
Yeah.
So a visual.
Basically, reductionism has been very successful in the past in terms of establishing new drugs. But reductionism is really it's a good process for dealing with simple diseases. But the more sort of complex diseases that are facing us now, reductionism really isn't the only answer. I mean, there's nothing wrong with reductionism as an approach. The problem with reductionism is thinking that it is the only solution. Because the problem with reductionism and breaking the thing down into smaller and smaller parts is that if you go down too far, you start to lose sight of what the overall picture and overall problem is. So in order to deal with more complex diseases, you need to have a better understanding of the whole system. And in order to deal with these complex diseases, you can't just use one drug. You have to use a combination.
Right. You've got in mind cancer.
Well, cancer, not just cancer, but pretty much all of these complex diseases. There's no single cure. There's no magic bullet. You need to use a combination in order to have the best effect.
Right.
So cancer really is an example of a really good example of a complex disease. I'm on slide. I'm on the next slide now, and what this slide basically shows is that the way you think, the way you behave, environmental factors, your genetics, your work, your diet, infections, and you read in the paper, every day, something's good for you, and then the next day something's bad for you, and cancer really is very complex. But despite that, there is an underlying simplicity, but only if you rise above the actual detail.
Okay, on the next slide.
So the other thing about cancer is cancer is a systemic disease. It's a disease which can affect all parts of the body. And
cancer is an infection then.
Cancer can be promoted by infections. But let's not go into that just yet.
Okay.
So moving on to the next slide, and we're starting to look now at what makes cancer happen, basically. Cancer is caused by cells that become selfish. We're complex organisms, and we're made up of millions and millions of cells, and all of these cells have to work in harmony. But in the case of cancer, something goes wrong, and an individual cell starts to basically take over the neighbourhood.
Right.
It's a noisy neighbour.
It fails to respond to signals to switch it off.
Basically we'll move on. That's on the next slide, actually.
Right.
So one thing I'd like to point out on this particular slide is there's a simple cross section of a cell. You can see it's almost like an eye. The centre of the eye is where the nucleus is. The outside of the cell, there's a big circle on the outside, and this is where the cell wall is. And what happens is there's receptors on the edge of the cell which basically sense the environment external to the cell. It takes signals, and those signals are then transferred down into the nucleus and change the way that the DNA is working. And what happens is, responding to the environment, the cell will manufacture chemicals and proteins to react to circumstances. So if we move on to the next cell now, the next slide. Now this is a slide which is it's